The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 5th episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian, and I've been in the flow cytometry space for over 12 years.
Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas with an additional location in Europe, we're both here in Philadelphia and our producer is in Missouri, so we're truly all over the map. We're ecstatic to have you listening to the 5th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcasts, you'll likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources and a focus on a topic of our choosing, before discussing any feedback we've received from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here, and we are looking forward to a fun episode today. To kick us off for episode 5, Brian is going to go over the news and resources before diving into today's topic. Brian?
All right. Thank you, Adam. We'll start off with the latest medical research news. And for today's stories, we're looking at 2 different cell therapy stories. The 2 trends that we've seen recently are sort of an expansion of what cell therapy can do in the oncology space, and then also the expansion of cell therapy outside of the oncology space as well. So appropriately, the first story that we'll be covering today is the recent FDA approval of the Iovance therapy, Amtagvi. And this is the first T-cell therapy for a solid tumor. Technically, it's using tumor-infiltrating lymphocytes, and it is the first one to reach the market. So we're incredibly excited about what this means. A lot of the cell therapies to date have been used to target only circulating tumor cells in the bloodstream. So this is the first one that's really going after, effectively, a solid tumor.
Right, and so you mentioned using TILs or tumor-infiltrating lymphocytes. Essentially, by utilizing a different cell type, they have the ability to attack a solid tumor, whereas your standard CAR Ts don't have that ability, or at least to the date in the clinic have not demonstrated that sort of ability. And so with this, it looks like targeting a specific mutation here, a BRAF, and there's a V600 mutation, right? And what this does is basically the tumor starts to grow because it's a whole serine, threonine, protein kinase, intracellular growth signal sort of thing that gets mutated and activated. And so trying to destroy the cells there because it's so over expressed, right? And roughly 50% of melanoma tumors have this mutation. I was interested to see, so because of the fact that it's a TIL, it's actually a bit longer to get this therapy out. The turnaround time is about 34 days for a TIL. My understanding is a little bit lower for your standard CAR T, although the price is somewhat in the same ballpark, or at least expected to be in the same ballpark. It is a one-time treatment. And it is derived from the patient's tumor. So like you said, very interested to see how this performs or continues to perform, because it is very much an unmet need in solid tumor.
Absolutely. And it's one of those things that will keep expanding where we can use cell therapies. So if you think about circulating tumors, that's almost always an extremely bad situation where the tumor is essentially metastasizing or the tumor is in the bloodstream itself, because this type of cancer can go to a wide range of different organs.
Hmm.
Being able to treat in a solid tumor can rapidly expand not only the types of tumors that we can target, so you can think about targeting a tissue-specific tumor as opposed to a blood-related tumor, but also the stage of cancer. So that could move cell therapies up from a very late-stage treatment up to a more early stage, first or second line treatment in some cases. So definitely something that we'll continue to monitor and continue to track as both Iovance and other cell therapy companies are pushing more into the solid tumor space. The other story, as we mentioned, was actually moving out of the oncology space entirely. So trying to use cell therapies to treat autoimmune diseases. And we've definitely seen an increase in the number of companies that are targeting this. And today's article is actually a paper. It is a sort of review or opinion paper, but it is indeed not a standard news article. So it is just reviewing some of the recent attempts at treating things like lupus, diabetes, and organ rejection, all of which are sort of afflictions where your immune system is dysregulated, and by adding some CAR T or some other type of cell therapies, you can potentially correct the imbalance in the immune system that's causing the autoimmune reaction.
Yeah, it's funny because at first when you had sent over this article, I was thinking more a specific cell type or something that we're utilizing here, as opposed to the normal CAR modality. However, some of the items that were discussed are really just looking at targeting autoreactive B cells. And so you still have your same anti-CD19 CAR cells, and you deplete the B cells. It generally takes, I believe it's a couple of months to restore the full the complement of B cells. But even much longer after that, patients have demonstrated, resolution, reduction in symptoms for these autoimmune diseases, like you mentioned, SLE, RA, multiple sclerosis.
Correct. And the symptoms and in some cases even the causes of these diseases are pretty widely varying. So it can be difficult to really attribute a single source of some of these things, or then also treat all of the symptoms that are in some way related. So having this type of really complicated treatment could help address a lot of these all at the same time, because these cell therapies can be extremely multifunctional and have a wide-ranging impact on the immune system. Mm-hmm. So definitely excited about what this means for the future of immune treatments and what this means for the patients currently suffering from these diseases.
Yeah, and I personally was really excited to see this come across because my thesis for my PhD was looking at alternative uses for chimeric antigen receptors. And at the time I was thinking about autoimmune, but looking at different cell types. And so here, this is basically just CARs, anti-CD19 CARs, but utilizing it in autoimmune space, and definitely a huge unmet need. I remember looking at data for RA, and patients that relapse and are resistant to treatment, and need to go from one treatment to another treatment, and it's very difficult. It's very painful, and so hope is that this can provide some relief there.
Sure. And the hope too is that we can help improve the safety profile and the cost profile to help treat some of these less than lethal diseases where possible, and that can help drive some of the innovation in the oncology space as well. Because as you had mentioned, with the last treatment, we're looking at costs up to $500,000 per treatment. Mm-hmm. Yes, it is generally a single treatment. However, that is just an incredible amount of money to pay for any type of treatment, even if it is life-saving. So definitely to improve the access of patients to these treatments, that'll be key. And I think one of those things that could be driven through other indications. All right. And I guess we can move now from the medical research and in terms of what's possible over into the investment news in terms of what different companies are doing within this space. And definitely the story of the day is really all about GLP-1. And the two different aspects of that GLP-1 story that we're covering today is the recent acquisition of Catalent by the Novo Nordisk, so the company behind Wegovy. And the other one is how the Roche team is expanding its obesity portfolio through some pretty significant acquisitions with Carmo. And both pretty significant stories there. Catalent was one of the largest CDMOs in the world. So for a major pharma to basically just acquire that company outright was a pretty significant development there. That deal total was $16.5 billion and really just goes to show exactly what the impact and what the need is for Wegovy in the market currently.
Right, and the expectation is, I mean, there's a huge unmet need looking at obesity as well, and in addition to type 2 diabetes. I was certainly interested in reading up on this. Not having personally been impacted by diabetes myself, and had read a long time ago about it. Really wanted to understand a little bit better. And so, looking at how GLP-1 it plays a role here, essentially what we have here, the GLP-1 agonists are mimicking the situation after eating, and GLP-1 is released. And so you stimulate that insulin production. And then, going through some of the tests, I was really curious, as you always hear on TV, A1C, and, what is that? And for myself, not personally, haven't looked it up. It was really cool to understand how the test works, that it's the glucose bound to hemoglobin, and over the course of time, the more you have on there when you're doing the test, you can actually get a snapshot of the last 2 to 3 months and understand if your blood glucose level is significantly higher. And so these type of medications are then going to reduce the amount of circulating glucose, by essentially mimicking GLP-1, and so providing relief in that way.
And the story with Wegovy and other similar GLP-1 type medications, has just been an absolute explosion of demand, both for diabetes treatments as well as just obesity treatments in general. And that's really driving Novo's appetite for acquiring a huge manufacturing organization. They're being exposed to all sorts of antitrust considerations based on how many other pharma companies were using Catalent for different projects. Mm-hmm. So to take on that level of risk and also just make this level of acquisition, it sort of gives you an idea of how significant this demand is. It's akin to America Online taking up the entire ability to produce compact discs for a couple years during the 90s as well.
Yeah, it's interesting, and there are multiple sides to this, right? And so they're purchasing the manufacturing organization to be able to produce, rather than building out all of this footprint on their own. Some other places have chosen to go the opposite. When there have been mergers, maybe one of the company that was purchased essentially had plans to build out manufacturing on their own, but that wasn't part of the strategy of the purchasing organization, and so then they outsource it. And a lot of different ways to handle how are you going to produce when, especially Novo Nordisk in this situation, expects to have a high demand for this therapy.
Sure.
So, and then the only other thing I saw, you mentioned about the Roche one, and again, we're talking about GLP-1, although their lead asset, CT388, is actually a dual GLP-1 GIP, so gastric inhibitory polypeptide, and so expected to work similarly in the way that the what we're talking about before, the GLP-1 agonist there. And this one is ready for phase 2 trials.
That's it. I think Wegovy has a pretty significant lead in terms of other similar competitors. But now that this business case has really been proven, I'm sure it will not be the only one on the market for very long at all. So definitely more to come in that space, and we have to figure out a good way to associate flow cytometry with all these assays as well, right? So definitely more to come on that side as well.
Mm-hmm.
All right. In terms of KCAS news for the week, the biggest thing on our plate right now is just the number of conferences that we've got going on. Definitely spring will be a very, very busy season. We've got the Boston Society for Cell and Gene Therapy coming up, as well as MetroFlow Vendor Day, the Cyto Conference, always a big one for flow cytometrists, AAPS WRIB, which is a lot of initials to cover sort of how bioanalysis gets done, AAPS NBC, which is essentially a very similar group on the West Coast as well as the World Vaccine and Immunology Conference.
Yeah, and so you're going to the one, the BSCGT, you said? That's in June?
So it's BSCGT, and it's actually at the end of March.
Okay.
Coming up quickly here. And then MetroFlow is in April. Cyto, AAPS, both WRIB and NBC are all in May. And I think World Vaccine and Immunology is in April as well, if I remember correctly.
Yes. And that one's in Washington, D.C.
Correct. So first of all, for any of our listeners, if you're at these conferences, please do come visit us. We'd love to talk to you about flow cytometry or any of the information you've heard on our podcast. And otherwise, look out for our presentations and posters.
Yeah, always great to make a connection, even just listen to the podcast, or, if there's something that you're interested in talking to us about in the world of flow cytometry.
All right. And then we can move on into the paper of the month, which is directly related to our topic here. And the paper of the month is Flow Cytometry and Cell Sorting, and it's a review by William Telford. It was published in the Frontiers of Medicine at the end of 2023.
And it's got an impact factor of 9.927. So definitely a strong impact factor there. Frontiers in Medicine, been around for a little while there. Really thought that this paper was a great primer for both the history of sorting and the technical aspects that are associated with it. I will say that we've talked about in our intro several times that some of my intro to flow cytometry was actually sorting on a Fax Jazz by BD. And I remember looking at that machine, having only really done basic flow, a few colors on a C6 Accuray or Accuray C6, and wondering how I was ever going to figure it out. One of the keys about a machine like that is that it's very much a manual machine. Everything that you have to do, it's not automated. Whereas a lot of the newer machines, basically it'll set it up for you. And there's some input, don't get me wrong. Obviously you have to understand flow cytometry. You got to understand sample setup, general machine setup and all, but drop delay, and laser alignment, things of that nature are generally taken care of. And so here, this paper talked about, the history behind it. One of the things that I like that they pointed out, so cell sorting, also known as FACS, so FACS. I'd heard a lot of times where people just refer to flow as FACS, and FACS actually stands for fluorescent activated cell sorting. And so definitely distinct from just flow itself, data analysis in that respect. And so, Brian, what stuck out to you in this paper?
What I liked the most was how carefully he handled the handling of the cell during the sorting process itself. And, a lot of people will hear, oh, you can sort cells with flow cytometry. And they'll get this picture of a cell being sort of gently placed from one side to the other.
Mm-hmm.
And what this paper really went through in the review was how violent this process can be on a given cell. So it went through the jet and air system. It went through the electrostatic system, and as well as some of the newer either microfluidic or piezo-based system. And that really just gives a good picture of the forces that the cell is experiencing. So you can think about, if your cell is more fragile or it's larger, let's say, then it's going to be subjected to these forces and may not make it through the other side. And that sort of explains some of the viability issues that are occasionally seen with flow cytometry cell sorting. And then also some of the sterility issues that you may, assume are there as well. If you're shooting this cell through the air, then it's being exposed to anything that could be in the air, so it must be done in a sterile environment if you want it to be like the cell that comes out to be sterile. And how the microfluidic systems, to some degree, get around that by having everything enclosed.
Right. And, talking about speed, right? So you can sort upwards of 30,000 events a second. This is more specifically jet and air, whereas the chips are somewhat slower. And so I know there's a lot of interest that they had mentioned there on utilizing chips for a car, cell therapy type things. It's just that right now the speed isn't there to support. And so it definitely talked about coming technology advancements there. One of the things too that's key though, and they did touch on this, is there's really three corners of sorting, right? You can think of it as a triangle. You've got your purity, you've got your yield, and you've got your speed, right? But you can only ever have two out of the three of those. You can't have it super pure, high yield, super fast, right? And so you balance that out between, do you want it pure and fast? Do you want to have high yield and fast? And, any combination there. It's one of those things, obviously, when you first get into sorting, you want all three, but you've got to make some compromises there to be able to meet the goal of what you are trying to sort. And there they did talk about downstream. A lot of this is genomic analysis. We've done support for clinical trials where sending out for RNA-seq, and the technology is getting higher and higher, better and better, that the amount of sample is not nearly as much that's needed to perform these analyses as it had been in the past. And so, you can sacrifice some of that yield and do sorts much more quickly, but still maintain high purity there. So yeah, I really enjoyed it from that perspective, and touching on the history of my own personal history there, as well as the machine. So yeah,
Adam, they didn't mention getting shocked by any exposed elements on their cell sorters. I noticed that as well.
So it's funny that you say that, because I do remember with the Jazz, those electrodes, and you're very careful, but unfortunately you do a ton of different sorts, an accident happens, and you get those little black spots on the electrode from where the shock occurred. It sounds like you may have had something similar.
I don't want to comment due to liability issues with the BD team. However, yeah, it's always a concern there. Definitely, fax systems have come a long way, both in terms of their capabilities and their safety. Let's just leave it there. So definitely, it's a great paper. Great to go through the history and see what sort of the developments are. In terms of what we have today. I think our readers are familiar, especially if they have seen the paper, with what fax is, and there's the customary figure there that really covers that the cells are flowing through one by one. The fluorescence is detected, and based on that fluorescent level, the cell is diverted into an aliquot tube. And initially there were 2 tubes that were possible. Now we can do up to 6 tubes from a single sort. And we can also do single cell sorting. And that is definitely a focus of the paper. But the number of different cell sorters that are now available have definitely significantly increased. So there are the bulk cell sorters. We of course have the BD S6 in our lab that can do the 6-way sorts as well as the single cell sorting. But also in the paper they mentioned the NanoSelect, which is sort of a benchtop bulk system, and definitely more on the lower side. And then the Tyto, which is a microfluidic base, so everything happens in a chip and you don't need to worry about containment outside of that chip. And I don't think they got into things like the cell-wrapped air.
I'm glad that you brought that up, though. The difference between the electrostatic storage, jet and air versus the chips, and the number of markers. One of the things that they mentioned was you can't generally evaluate as many markers, and usually can only do one population at a time for the chip-based sorting. I know you've done some research on this recently. The paper isn't that old, so I would imagine that the information is still fairly accurate. But when you've done that investigation, is that still the case?
I think with the microfluidic systems, you can do a few more markers now, but certainly you do then need to potentially do multiple runs. So because the system is gentler on the cells, you've got that capability where you can run the cells through once and let's say get your CD3 cells, and then put another marker on and then run the cells through again and get your CD3, CD8 cells, and then run it again and get another marker on the CD8 subpopulation. So it is definitely possible, but as you say, definitely a much more tedious process compared to with the S6 or other sort of large scale sorters, being able to put 4, 5, 10 markers on a panel and run it through all at once to get a very specific cell population.
He said 4 or 5 or 10. We're well in excess of that with the S6 that we have, though. We've got the 5 laser unit. Don't forget that.
That's true. That's true. In theory, up to 30. I just would not want to be gating all 30 parameters there on the sorter day of. But with the proper preparation, I think we've done at this point 20 colors. So definitely a capability there.
Yeah, and it is good to highlight the differences there between the units. So when you are utilizing something like an S6 or a Jazz or whatnot, between each sort, each sample, you're going to have to clean the machine and get ready for the next sort. Whereas with the chip-based ones, you switch the chip, right? And so it is different there. The contamination, that's one of the reasons why I mentioned about doing cell therapy sorting, is because they prefer the chip just because of that as being one of the cores. Is you can just basically take the chip. You don't have to worry about all the cleaning and everything like that, and replace it with a brand new chip that had not been used, and so it is obviously sterile.
Sure. And I really am seeing that sort of separate into 3 different directions with the development of these cell sorters. As you say, definitely anything in the manufacturing arena, the chip-based reigns supreme because of that quick change capability and its contained status. But then on the research side, the vast majority of research applications, or like preclinical is what I'm speaking here, where we're looking at maybe is GFP in the cell line or not. But it's using very small benchtop type cytometers that are good enough for the purpose at hand. And then for clinical and CAR T applications, we're seeing a lot more of those higher parameter use cases as well.
Yeah, and I mean, one of the things that the units like the S6 excel at, I mean, you could do 96 well plate sorting and either put a specified number of events per well, all the way down to 1 event per well. You can do index sorting, and so you're able to take a look at the various parameters that you had analyzed on that event as it is sorted into the well, and so you can can get a snapshot. And one of the things that that's helpful for is if you're doing clonal expansion, you can take a look at the source material there and help you set up for future sorts, for example.
There's that, and then I'd say the killer application within clinical testing that needs that single cell sort is probably the single cell RNA sequencing.
Mm-hmm.
So I know we work with a couple of groups that are doing that currently, and then also I've seen quite a few papers that are publishing on the technique. Towards your point earlier, the amount of genomic information or nucleic acid information that you need to perform this type of assay has significantly decreased. So as long as you're sure that you're getting a one cell per well, you can definitely do these assays. That always comes with a caveat that the cell will not be a super happy cell after the stresses that we had mentioned at the beginning of the talk here.
Mm-hmm. Yeah, you definitely want to have happy cells. That's, I mean, down the road, if you're doing something like RNA-seq, it's I guess it's a very short period of time that it's going through that pressure, and then the cell is in a buffer, essentially a lysis buffer, so that it's ready for downstream analysis. You're not worried about the health of the cell post sorting, right? But then there are certainly ways that you can take it easier on the cell when you're doing sorting and you're not using chip-based. Chip-based just naturally has lower pressure, but you can make sure that you're stacking the deck in your favor when you are doing standard sorting there.
Sure. All right. Anything else that you would usually consider when you're starting out a sorting project?
So the starting out for a sorting project, you've got to understand your sample type. And so they did touch on the size of the nozzle. I know from experience, certain cell types, if you're trying to use the 70 micron nozzle, they're just not going to fit and they're going to clump up. And so you might want to switch to the 100 micron nozzle. And so it's certainly helpful to understand if your cell type is a larger cell or it's a smaller cell right at the outset. Then, obviously the normal flow parameters, what are the markers that you're looking at? What are you trying to sort here? If it's a heterogeneous population, what is the level of expression that you're expecting, representation within the cell types, and then biasing that too for how much of that population are you trying to get? Are you trying to get high expressor, mid expressor, low? Are you getting non-expressors? And so all of that is something that is important for your setup so that you're getting the optimal sample type. And then as far as the backup as well, you're looking at, are you just going into a tube and doing a bulk sort? Are you doing single cell sort or enrichments? One of the key considerations if you're trying to expand cell lines and you want to enrich is that there is drift when you do sorting of cell lines. And so you may sort and enrich for a high level expression, but those cells may not grow as rapidly as cells that express your marker at a lower level. And so over time, you may want to, let me take a step back. You may want to grow out your cells and bank them very quickly so that you have that original bank to go back to, rather than just continue to use that initial sort. And then at one point you find out it no longer is expressing the marker of choice as highly as you had wanted.
Interesting. All right. And I know that's more from a sort of a preclinical and more like a cell line generation approach. Mm-hmm. And then I think the other thing that we found with the cell therapy type approach too is if we're sorting the cells, and especially for RNA-seq, you had mentioned basically doing that as quickly as possible. So if it is going into the lysis buffer, then making that a relatively short process because those RNA signatures can change pretty quickly in response to stress. Right. So as you say, potentially lysing them immediately and then potentially freezing them immediately as well.
Right, exactly. Going back to the point about cell lines and enrichment, one of the ways around that is to do single cell sorting and do it into 96 well plates. You're not always going to get clones that grow out. Generally, though, if you do single cell sorting, if I remember correctly, I think it was probably about 2/3 of the plate I would have colonies growing out. Dependent on cell type, of course, there are some cells that enjoy crowding. And so what you might want to do is add preconditioned media into the wells. And what it does is have, various cytokines, other cell factors that are present there that somewhat mimic the presence of other cells within that culture and allow the cells to grow out. Other things that can be done are increasing serum as well, the percentage of serum, just to help them along there. So I absolutely. You can tell my history with sorting really involved in the details and some of the things that make you rack your brain day in and day out trying to figure out how to get these cell lines to grow. Because I think a lot of times when you're on study teams, the expectation is I can just transfect or transduce a cell line and I'm going to have my receptor of choice. And it doesn't always work that way. There's obviously biology of the receptor and the cell type that you're working with. And so, seeing things like expression drift and lack of cells growing out when you're trying to do clonal are certainly key considerations there.
You're saying that those, 4 steps in the protocol or the poster presentation aren't always, like a solid 1-week process? Craziness.
Usually more like 3 weeks.
Okay, all right.
To get it to grow out, but yeah, absolutely. And you mentioned about the RNA-seq. I know you're talking about a lot of the workflows that are pertinent now. Just making sure that we're able to confirm. So when you sort, you can always do a bulk sort and then test it to make sure that you've got the expression that you want. And that's definitely common. Trying to think if there's anything else. And to your point, just making sure you've got to freeze down things rapidly before there's any further impact to the expression in the cell.
Okay. And, one of the things that I'll be asking about as we go to all these conferences is, for our fans to write in comments to the show, but also see what either tips and tricks they have for their sorting workflows or what things they'd be interested in us talking about on a new podcast as well. So get ready, conference attendees. You're going to get a dose of the conversational flow and ideally be featured in our future episodes here. But with that, maybe we should move on to, sort of wrapping up the episode with weekend plans.
Let's start with you because I'm trying to figure out what my plans are for this weekend. I may end up just deferring to previous weekends as a recap of what I had done.
No worries. So my wife has purchased a unfortunate amount of paint, and it looks like I'm going to be doing touch-ups for most of the weekend. All interior, luckily, but it looks like it's going to be quite a task.
More than one room?
At least 2, if not 3 rooms. And every time she sees another room, she's like, "Oh, we should totally touch up this one last part as well." So devil's always in the details.
Mm-hmm, mm-hmm. Yeah, absolutely. Now, are these oil-based paints, or what type are we talking about?
I think they are. That is beyond my paint knowledge. My paint knowledge stops at, I think, flat or semi-gloss, and that's where I end.
Fair enough. I just have memories of using an oil-based paint to do ceiling one time. it just was kind of stained when we first moved in. And oh man, even in the middle of January, we had to have the windows open to be able to do that without passing out. We even had masks on, ventilators, all that kind of stuff. It's just still overwhelming. I'm used to using more of the latex paints, and so just a little bit less in the way of fumes and whatnot. So hopefully that's what you'll be using. For me, no paint plans for this weekend. Last couple of weekends, gone to Topgolf. Really enjoyed that. Did that with my father and my brother. And so my father's now, I think he's going to be 80, 81. And here we go to play Topgolf, and it's the first time he's ever done it. He's been a golfer his whole life, and he wins 2 out of the 3 matches. Beats my brother and I, so he's still got it, man. I hope I can say the same when I get there.
Are you going to call that beginner's luck, or is that just golf skill?
I think it was golf skill. He also so. Here you go. I'll give a little bit more feedback of the way that I play golf. My goal was really to see just how far I could hit it. I was taking a 3 hybrid and, hitting it out to the white cups there. So I should take a step back too and say that what this is, if you're not familiar with Topgolf, is they've got certain areas that you can hit a ball and the ball goes in. They track where it goes, and then you get points based on how close to each one of these cups and flags they have. And the further out that you hit it, generally the more points, especially if you can get it in the center of the further out cups. But, you're trying to hit it out far, you're going to hook it, shank it, whatever. He went with a shorter club. I think it was a 7-iron, and he was just dropping it in, right, the closer cups, and he knew what he was doing. He's like these young guys, and I say young comparatively here. They really, need to learn a thing or two from their old man. So he put on a show. And then before that, went to a Sixers game with some friends, and so that's always a good time. They treated us with a W over the Cavaliers. So
nice, excellent. Well, that sounds like a good one. And then next, we'll have to come up with some future plans. And I think for our next recording, also we're going to move into the receptor occupancies for our primary focus. So that could be a multi-part series where we talk about the biology or the validation or the analysis of those as part of clinical trials.
Yeah, looking forward to that. I mean, RO is something that's requested all the time, and a lot of considerations that go into it, but a very useful assay.
All right, excellent. Well, with that being the case, I guess we'll call it a wrap for this month, and thank you to all our listeners for joining in.
Thank you.